This is a recording for our Chapter 4 on viruses. The topics that will be covered in the two sets of slides are viral characteristics in general, and specifically for the first half of this will be on bacteriophages, and the second half will be focused on animal viruses with some crossover. There are key concepts for this unit, which for these sets of slides, actually for both of them, for the entire chapter, would be understanding, and you can change life to replication cycle, but being able to compare and contrast the lytic and lysogenic phases of a bacteriophage infection and the lytic and latent animal viruses. And for animal viruses, you can kind of add in there understanding the difference between a naked animal virus and an enveloped animal virus. And there is one other type of phage that we will cover that's not in your chapter, but I have supplemental information on Canvas, and those are the filamentous bacteriophages. So we'll look at all of those. So first, some questions here. What are viruses? You may ask yourself, are they organisms? Are they alive? And this one, to answer it, you really have to think back on some general biology information and how biologists characterize life. So we have 7 different characteristics that we say are the definition of life. And one of them is that the basic unit of life is a cell. And so that therein states that viruses are not living, because viruses are not cells. They have very little about them that reflects cellular organization, even the simplest prokaryotic cell. They're composed of proteins and nucleic acid almost extensively. For the envelope viruses, they gain a cell membrane from their host cell, but they don't make it. They don't have the information or the machinery to make anything themselves. So they are not considered alive. I just mentioned this one. What distinctive biological characteristics do they have? Well, they do have nucleic acid. Not all of them have DNA, which is one of the characteristics of life, replication through DNA. But they do have nucleic acid, and they do use the nucleic acid in order for replication to occur. So in that regard, you can kind of bridge living and non-living categories together. They also can respond to their environment. They have receptors that allow them to bind to they have glycoproteins that allow them to bind to unique receptors on host cells. And they can respond to environmental fluctuations within their host cell if they are integrated and essentially jump ship or excise themselves out. So they have a few features that are shared with biological organisms, but nonetheless, we still do not characterize them as living things. How can such simple particles cause disease? Well, I think most ready answer to this one is their astounding rate of replication. So a single poliovirus that infects a human cell can burst out as 50 million poliovirus particles, right? 1 to 50 million in a single replication event. So the sheer number of virion particles is really what enhances their pathology or their virulence. And what's the connection between viruses and cancer? At the end of the next sets of slides, and we will get to the answer to this question, at least a little bit of it. In general, the viewpoint is viruses are not non-living because they control host cells. So they do have the ability to manipulate their host cells. They get in, they release enzymes, and they either halt host machinery or essentially they just hijack the host cell and take it over. So this is the accepted statement. Viruses are infectious particles. So they're not cells, they're not living organisms, but they're infectious particles, and they can be either active or inactive. So we don't call viruses alive and dead. When a virus no longer has the ability to cause infection, we call it inactive, but we do not call it dead. They are all obligate intracellular parasites. We already know what this term means, but just as a refresher, obligate is requires, intra is inside, cellular obviously, and then a parasite from our chapter 6 on ecological associations. This term means one benefits at the cost of the other. And this term parasite also indicates a symbiotic relationship where one requires the other for survival. In this case, the virus requires the host cell for survival. There is a known virus that can infect every type of cell. So it's not true that any virus can infect any cell, but there is a known virus associated with every known cell that's out there of any type of organism. They're very, very small. So, for example, 50 million polioviruses can be inside a single human cell. And this is a picture to show that relative size comparison. So here's a yeast cell down here. Here is a smaller eukaryotic cell, and then some of these regular bacterial cells that we've been seeing regularly, including E. coli and Streptococcus. Rickettsia is one of the smallest known bacterial cells, so 0.3 micrometers versus Streptococcus that you guys have been looking at, which is 1 micrometer in diameter. Here's the largest known virus, which is the mimivirus at 450 nanometers, and all the rest of the viruses fit within this category too, so between 150 to 20 nanometers, and this is 1 times 10 to the negative 9th, so very, very tiny things. In fact, the smallest virus here, yellow fever virus at 22 nanometers, is only a little bit less than 10 nanometers bigger than the largest human protein, which is hemoglobin, right? So it's a really, really tiny particles that are still able to cause infection. And this picture is nice because it also shows you some variations in these virus organizations. We will be discussing only bacteriophages and animal viruses in our conversation, but please note that there are viruses that infect every living organism. We've mentioned this in class already, but these amazing mosaic tulips, this pattern on these tulips is due to a viral infection. And in fact, when you remove the virus from these plants, then they no longer produce this really cool pattern. Here's a picture of Ebola as a classic example of a filamentous animal virus. And this is a picture of something like E. coli that's being infected by each of these individual blue things, which are complex bacteriophages. So this is a virus, this is a viral infection, an ongoing viral infection. So some general structures and general characteristics that are shared by some or all viruses. This is a picture of 2 different types of animal viruses. So the top here is a naked animal virus, and here at the bottom is an enveloped animal virus. And for both of them, you notice that it has the same basic core unit. And this is composed of a capsimer or a capsid head. This is made of capsimer subunits here. And inside is nucleic acid. And the 2 of these things together, the capsid and the nucleic acid, is called the nucleocapsid. This is a term that you should become familiar with, the nucleocapsid. Now you know that there's also a nucleocapsid here on an enveloped animal virus. In this case, the capsid, or the capsimer subunits surrounding that nucleic acid, are encoded in this giant envelope, which is derived from the host cell upon budding. So this is an exocytosis, or budding, off of a host cell. This is actually a phospholipid bilayer from a eukaryotic cell, and you'll note that it has a bunch of virus proteins that are all embedded in it, including some additional matrix proteins that are found on the inside of that membrane. Now, both of these things share this extra feature here, which are glycoprotein spikes. These spikes are either a part of that capsid for these naked animal viruses, or they're embedded in the cell membrane for these enveloped viruses, and this occurs right before release for these enveloped animal viruses. Now, the importance of the spikes: these things are glycoproteins, and they have high degree of specificity towards very particular receptors on the surface of their host cell. So the spikes are what's involved in the binding and attachment to the host cell. But you notice that every virus has a capsid. So this is true of all animal viruses, plant viruses, bacteria viruses, etc. And this capsid is made of these capsimer subunits. Some animal viruses, as mentioned, can have these envelopes. This is not true of bacteriophages. They do not carry an envelope with them, and this is in part due to the unique nature of the bacteria themselves. Right? There's an extra cell wall layer that really prevents the ability for this envelope to be budded off, essentially, from these viruses. And then animal viruses have these glycoprotein spikes. Bacteriophages have glycoproteins as well. They're just slightly different in nature. The core can either be DNA or RNA, can either be single-stranded or double-stranded. In addition, some viruses carry with them extra proteins like matrix proteins, as mentioned here with this envelope, or extra enzymes. This would be really important if a virus comes in with a form of nucleic acid that our cells or their host cells cannot process, like for example RNA, and they have to come in with their own enzymes that can convert that RNA into DNA so the host cell can actually process that. So we'll look at each of these in a little bit more detail here. Again, the capsid is composed of capsimer subunits, and this varies depending upon if we're talking about these geometrically arranged icosahedral viruses. These have a very specific arrangement of their individual proteins to form these icosahedral heads, and you can see it with this electron micrograph of a naked icosahedral virus. You can see these glycoprotein spikes here. Versus a helical or filamentous virus, which is depicted here in this electron micrograph. In this case, the capsimer subunits are arranged in this helical formation, and in fact, the nucleic acid is sort of wound up within this virus itself. The organization of those capsimer subunits provides the shape of the virus itself. So we were just looking at this icosahedral versus filamentous. So you can find for animal viruses, these are the two primary organizations or arrangements of animal viruses, the icosahedral and then this helical or filamentous virus. An example of this is the tobacco mosaic virus for plants. I've already given you an example of the Ebola virus, or any of those hemorrhagic fever viruses would all be this filamentous form for humans. And adenovirus, which causes a common cold, is a really classic icosahedral form. For bacteriophages, they can have also this filamentous form, or they can have this really, really unique complex arrangement, which contains this icosahedral head, or the nucleocapsid, with the icosahedral capsimer arrangement and a nucleic acid, but attached to it is this entire complex tails. So we have the sheath here, and let's look at another picture of this. So we have the sheath here, a collar part here, tail pins that are involved in binding to and penetrating through the host cell envelope, and then these really cool tail fibers. And these tail fibers are where the glycoprotein spikes are, right at the tips of these things, not spikes, but glycoproteins that are involved in host cell attachment and recognition. So still glycoprotein components here that are involved in host cell binding, but a little bit different where they're placed. This is an electron micrograph of a complex bacteriophage. Excuse me. Animal viruses, as mentioned, can contain these envelopes as well. As mentioned already, they are acquired upon exit from host, and we'll look at how this happens in a little bit more detail shortly. This, as you should note, has those glycoprotein spikes that are embedded in the envelope itself, and the envelope surrounds that nucleocapsid on the inside. The function, why organisms or why some viruses may take this capsid or this envelope, actually for both the capsid and the envelope, both of them provide protection from environmental degradation of their nucleic acid. They're both involved in binding and penetration, but unfortunately they both are also what stimulates that immune response. It would be, maybe you would think that the envelope provides some protection from the host immune system because it's the host cell, but because the virus embeds all of that envelope with virus proteins, the proteins themselves are immunogenic, and so they will still stimulate that immune response. Inside all viruses is nucleic acid. As mentioned, this could be either DNA or RNA. This could either be single-stranded or double-stranded, and this is the only place on the entire planet that's known to have double-stranded RNA. So this is really, really unique. RNA is usually produced as a process of transcription, and it's produced as a single strand. single-stranded molecule, but some viruses exist as a double-stranded RNA virus. This is so unique to viruses that all mammalian immune systems have evolved an antiviral defense to this one particular molecule, to double-stranded RNA. So it does not, the host cell, like our bodies, do not need to know that we have a virus inside. If we detect a double-stranded RNA, we will immediately send out antiviral defense system, because this is such a unique component of viruses. All viruses have very, very, very small genomes. This is between 4 and 100 or 4 and hundreds of genes. This is really tiny compared to any other, even a prokaryotic cell. And these genes, right, the smallest genome has genes for getting into the cell, so invasion, redirection of host machinery, and then the structural genes, so those capsimer subunits must be encoded, and then any of those extra enzymes that are needed for reverse transcribing, for example, the nucleic acid, or perhaps those matrix proteins for the cell envelope. There we go. So some have enzymes for specific operations like replication, etc. The glycoprotein spikes, again, these glycoprotein spikes are found on animal viruses, but remember that glycoproteins are still found on the tail fibers of bacteriophages. And for both bacteriophages and for animal viruses, this is what provides that host specificity. So they bind to host cell membrane proteins, or cell envelope proteins for bacteriophages. This confers a very, very high degree of specificity. Even within a multicellular organism like a human, viruses can be very specific to one cell type, and they cannot bind or infect any other cell type that doesn't have that one particular receptor. This provides a very narrow host range for each virus. Overall, for all viruses, they need an actively metabolizing host cell. Remember, they are obligate intracellular parasites. They cannot exist outside of the host cell. The general process, for all viruses, the virus either enters into the cell or releases its nucleic acid into the cell, and the cell then processes the nucleic acid, replicates it, transcribes and translates it into more virion proteins. Those proteins then spontaneously assemble into virion particles, and then those particles are released. This slide is just to show you this variation depending upon which type of nucleic acid that virus comes in with. If a virus has double-stranded DNA, it's in luck, because all eukaryotic and prokaryotic cells also have double-stranded DNA, so our cells know what to do with this. We will transcribe it into mRNA, translate it, we will also replicate it. If, however, a virus has mRNA, a positive sense strand or a negative sense strand, if it has any other variations of it, like a single-stranded DNA, then it must have its own enzymes to convert that form of nucleic acid into a form that our cells can recognize and process. The most classic example of this is this process of reverse transcription. HIV has this, as well as other viruses, where they come in with one particular form of messenger RNA, a single strand of it. Our cells cannot replicate this, and so in order for that virus to gain replication of its nucleic acid, it comes with its own enzyme called the reverse transcriptase. These are called retroviruses, ones that have this process here, this reverse transcription, where their own enzymes are required to reverse transcribe mRNA into DNA. That DNA is then essentially replicated and transcribed to make more copies of this viral nucleic acid. So for virus-host interactions, let's start with bacteriophage infections. And there are two different ways to look at the type of infection that a virus can do or can perform, and that would be either productive or latent. Now, productive infection is one that results in active virion particles at the end of an infectious process. And for this one, for bacteriophages, your book really just talks about the lytic phase in bacteriophage infections, but I will also introduce extruding phages. These exit the cell through a process called extrusion. These are filamentous phages, and they're non-lytic, so they keep the cell alive, but they produce active virion particles during the process of an infectious cycle. Now, these are in contrast to latent infections. Latent infections, there's a lot of vocabulary terms for you here, but these ones undergo periods of latency, which is also called periods of lysogeny or the lysogenic phase in a temperate phage infection. So for these terms here, the type of virus or the type of bacteriophage that is able to undergo the lysogenic stage or the lysogenic phase of an infection is called a temperate phage. So not all viruses are able to do this, but temperate phages are able to enter the cell and enter into latency. When the host cell is infected with a temperate phage that's going into this period of latency, the host cell becomes the lysogen. So it has been altered by the introduction of temperate phage DNA. The lysogenic stage is what we're calling this, or the lysogenic phase for a temperate phage. This is where viral DNA is integrated into the host cell chromosome. Now this last term here, lysogenic conversion, this is a term that we use to describe the alteration of the host cell, so the bacterial cell, when the phage DNA is integrated in the host cell chromosome, and that integration leads to the production of new proteins in the host cell. In other words, a conversion of the host cell based on the phage DNA. Okay, so that's lysogenic conversion. This picture here, I like it because it shows you everything we just mentioned in a more visual format. So again, these viruses or virion particles cause an infection. And for bacteriophages, that just means a virus injects its nucleic acid into a host cell. That's what this part means here. Now, once that process occurs, it will either lead to one or the other, right? So either it will lead to a productive infection where more virion particles are produced, or it will lead to a period of latency or a lysogenic stage where there's either no effect to the host cell, or the host cell can have genetic alterations where the host cell is modified. For over here, the productive infection, there is either a death of the host cell with lysis. This is the release of virion particles. This results in the death of the host cell, or a continuous release of virion particles that allows the host cell to stay alive, and a continuous leakage of virion particles. Over here, this period of latency, this is the lysogenic phase of a temperate phage, and we'll see pictures of that in just a minute. This is where the nucleic acid becomes integrated into the host cell DNA, and once it's integrated, it just becomes a part of that host cell. So it's replicated. It can be in the form of a plasmid, and in this case, it's also replicated, but it's independent of the host chromosome. We'll learn more about plasmids and how they work in Chapter 8. In this case, the host cell is often modified, but it does continue to multiply. So we'll start with the lytic phage, and let's look through the process of this replication cycle. For lytic phages, this is a picture of a complex bacteriophage infecting a host cell. In this case, you see that host cell is just about to burst. This is a lytic productive infection. Most of these things have double-stranded DNA, so it's a very quick and easy process for replication and synthesis. The T-even phages, so T4, T6, T8, etc., these guys are the most readily studied just because they infect E. coli, which is the most commonly studied bacteria. This is the replication cycle: attachment, penetration, synthesis, which is transcription, translation, replication of replication of phage nucleic acid, spontaneous assembly, and then release through lysis. Let's look at these in step-by-step format. So step one is absorption, and we're going to kind of go back and forth, and we'll make a little chart here. So for this lytic, and I put here the lytic phase because this is with a temperate phage. They can switch between a lytic phase and a lysogenic phase, but this, you can also call them lytic phages. So step one is attachment, attachment or adsorption, and this is through tail fiber glycoproteins. And there are various modes or sites of attachment on both gram-positive and gram-negative cells that these tail fibers can bind to that host cell envelope. For gram-negative cells, this could include the porin proteins. It could actually be a part of that lipopolysaccharide, so the O antigen. or the sugar, the polysaccharide in the lipopolysaccharide layer. For gram-positive cells, oftentimes it would be the teichoic or lipoteichoic acids. This could be the fimbriae, this could be a part of the flagellum. So there are many different sites of attachment or absorption for these bacteriophages. Note that as soon as there is attachment or absorption of these tail fibers, the tail pins, which are these physical structures that essentially embed themselves into the host cell envelope, there's oftentimes an enzyme that's also released once you have the tail pins embedded to help burrow a hole through or penetrate through that cell envelope, because passing through that cell membrane is very challenging. Once absorption occurs, then we have penetration, and in this case the bacteriophage DNA or RNA. In this case, since we're talking about lytic infections, this DNA is literally injected into the host cell. So for step 2, this would be penetration, where the phage DNA is injected into the host cell. And just note that that means that the bacteriophage structure is left outside the cell for bacterial infections. Step 3 is replication of phage components, replication of viral genetic materials. So you have replication and synthesis. So synthesis is the transcription, translation of viral nucleic acid to make new proteins that are then spontaneously assembled and then matured and then released. So for us, we'll put step 3, and these step 3 and 4 are kind of interchangeable, but we'll put replication, phage DNA with host excuse me, with host cell machinery. Step 4, synthesis of phage proteins, synthesis, which means, transcription and translation using host machinery. Excuse me. Spontaneous assembly, we'll see another picture. This is not as spontaneous as it seems. There is affinity for these proteins to bind to each other in sort of a step-by-step fashion. Oftentimes there are extra enzymes that are encoded by viral nucleic acid that assist in this synthesis process. So it's not necessarily just random assembly to generate these beautiful virion particles. There sometimes is organization to this. But nonetheless, we have spontaneous assembly of virion particles. Maturation means that they come together. The nucleic acid is packed into those capsid heads to form those nucleocapsids, which are attached to the rest of the structure of the virion particle. And these guys are ready to be released. So maturation and then release is through lysis. So for this one, we'll just put release is through lysis. This is another picture of the same process. So step 1 here, attachment or absorption. Phage attaches to the host cell through these tail fibers. And again, it would be very specific spots on that cell envelope. And it's definitely not the cell wall. Cell wall is protected in gram-negative by that lipopolysaccharide layer. So there are various modes or spots on that bacterial envelope that are sites of attachment. Once they attach, those tail pins burrow through that envelope, and then you have penetration of that nucleic acid. That nucleic acid is then transcribed and translated into phage proteins. It's also replicated so that there's more copies of that nucleic acid. Now we have synthesis of the proteins, spontaneous assembly that occurs, and as mentioned, it's not as spontaneous as it seems, so there is some organization to this assembly process where that nucleocapsid is created and then is assembled with the rest of this, the rest of the body of this virus to produce this mature virion particle. After spontaneous assembly, then you have release through lysis. This is just another picture to show this penetration step here, where that head essentially is just emptied out with that nucleic acid secreted into the cytoplasm. And this is a true picture. It's an electron micrograph of a bacteria that is actively being infected by virion particles, but specifically by the nucleic acid of the viruses. This is another picture just to show you the chaos of this. There's no, we call it quorum sensing for bacteria to sense each other. There's none of that for viruses. So they have no ability to detect the presence of other virion particles that are next to them. So they will continue to bind and inject their nucleic acid into a host cell, even as that host cell is lysing open. So that was type 1 type of productive infection was a lytic infection. A second type is extrusion. This is, again, a productive infection leading to these virion particles being generated at the end of an infectious process, but in this case, it leaves the host cell alive. These are always with filamentous phages, so they have this long helical arrangement. The replication site is through the tip of the F pilus, which is otherwise known as the sex pilus. That's what we've discussed it as. When we get into our chapter on genetics, you'll see why we call this the F pilus. It stands for fertility plasmid. But in any case, this is the actual pilus structure that extends out of that cell. This does not completely abolish the host cell function. In fact, that host cell stays alive. And we'll see how the capsular subunits align the host cell membrane, and so that you have assembly during the release of the cell. So here's a visual picture of what's going on here. So here's the beginning. We have a filamentous phage that binds to the tip of the sex pilus, or the F pilus. This is a close-up picture of what this filamentous phage looks like, where these capsular subunits essentially wind themselves in helical arrangement around the phage DNA. In this case, the filamentous phage binds to the tip of this F pilus and then injects the nucleic acid through that sex pilus. And so once this nucleic acid is inside this host cell, the host cell will, just as before, replicate that nucleic acid and also synthesize new virion proteins from the genes that are on this nucleic acid. So we also have synthesis, transcription, translation of phage DNA, but replication of phage DNA. But because these guys don't lyse the cell, they stay inside, and that means that host cell, during its regular binary fission process, will just naturally pass on that phage DNA to the progeny. And this transmission here from host cell to daughter cells is called vertical transmission. So not only do we have productive virion particles able to infect neighboring cells, which is horizontal transmission, but we also have an example here of vertical transmission. Now for extruding phages, these guys do not lyse the cell, but instead you can see here that the capsular subunits are synthesized and then they're put essentially as a lining on the inside of that host cell membrane. So upon release, these filamentous phages, the phage DNA moves to the cell envelope, and then it can leave that cell, usually through an enzymatic reaction that kind of generates a hole in the cell membrane, but during release, those capsular subunits just wind themselves around that nucleic acid, and essentially that's how this phage is extruded or pulled out of that cell. But it keeps that host cell alive. So the host cell is now called a carrier cell in this regard. So let's add these to our list here. For extruding or filamentous phages, step number 1. Why does it say 2? Step number 1 is attachment, adsorption. But in this case, we do have these filamentous phages attached to the F pilus. Let's put sex pilus. 2 is penetration. Same thing, phage DNA is injected. 3 is same as before, replication of phage DNA. 4, synthesis of phage proteins. But 5 is also a difference. So for 5, the capsular proteins line inside of cell membrane, and so that 6 release is through extrusion. Okay, release is through extrusion. But again, it is an active infection. So this discussion question is one that you guys can go over, take a few minutes and think about a few different ways that extrusion can give advantage to a phage. I guess I could kind of answer this one here too. So for phage extrusion, we talked about this a little bit in class, but certainly there is protection that is offered once this phage DNA gets inside this host cell. That DNA is no longer susceptible to environmental degradation, so it's protected from heat, from radiation, etc. This also provides a secondary means of transmission, so not just vertical or horizontal transmission, which is the normal process of infectivity for viruses in a population, but now also a vertical transmission here, so that's number 2. And then number 3, of course, that host cell just stays alive, so it's an ongoing productive infection. There's no real end to this infectious stage. Okay, and the third type of bacteriophage infection would be the lysogenic infection or the lysogenic stage for a temperate phage. In this case, we are also dealing with DNA viruses. These guys undergo the process of absorption and penetration, just as we saw before with a lytic bacteriophage, but instead of progressing into a productive infection, after penetration, that DNA for the virus circularizes and then integrates itself right into that host chromosome. So this is called splicing, where essentially that host chromosome is split open and that viral DNA just integrates right inside. This is called the prophage stage. When it's sitting inside that viral genome, it's called a prophage. Here's a picture of it. So again, absorption, penetration, just as we saw before with the lytic cycle. But whereas in the lytic cycle, we have replication of phage components and a generation of more virion particles, here we have the period or the stage of latency, where we have integration of viral DNA for an indeterminate period of time. So this prophage stage could just stay forever, potentially, right? Or at some point, this viral DNA could excise out and lead to a lytic infection. So let's add this to our little list here. So this is going to be exactly the same. Steps 1 and 2 are the same. Of course, my numbering is off. 1. Sorry. 2. Okay. Now 3 is, this is the prophage stage. Integration of viral DNA into host chromosome. And again, this could stay indefinitely in this prophage stage. Now, at some point, if that host cell becomes damaged, if there is environmental stresses, if there's extensive UV damage, etc., then this lysogenic phase could end. So if there is damage to a host cell or there's other environmental trigger, then you can have the excision of prophage. And if you do, this will lead to a productive, or I would say a lytic infection. So if this does happen, you'll have this DNA cut out and essentially move right back over here to steps 3, 4, 5, and 6. And I think I have a picture of how that would look. Yep. So here we have an example of phage lambda. This is an example of a temperate phage that can undergo both a lysogenic phase and a lytic phase. These temperate phages prefer to enter into a period of lysogeny and not to generate an infectious a productive infection. So in this case, we have injection of phage DNA, and here's a picture of that phage DNA, that linear DNA that circularizes, and then we have that integration that we saw before in that picture, and this is called the prophage stage, right? The prophage is integrated into the bacterial chromosome. In this case, the bacterial cell is really not altered, or potentially could be modified slightly by gene expression changes based on genes that are encoded on this prophage DNA. That would be lysogenic conversion, which we'll see a picture of in just a minute. But in this case, the host cell stays alive, it continues to replicate just as normal, and during its normal replication process, it also replicates that integrated DNA. And so we have herein another example of vertical transmission, right? Vertical transmission of this prophage DNA in this population. And the cells continue to multiply. If however, there is damage or stress to that cell, you can potentially have excision of that phage DNA, and that would lead to that phage DNA being excised out or cut out of the chromosome, and once it's released, it will progress into a lytic infectious process. So this is the lytic cycle, where we have, again, step 3, 4, 5, and 6, replication of phage DNA, synthesis of phage proteins, spontaneous assembly of new virion particles, maturation, and then release through lysis, right? And so then this process can begin again. These guys could potentially, instead of undergoing this integrated pathway, the lysogenic cycle, here at this step after penetration of nucleic acid, could potentially progress directly into a lytic cycle, right? So there are some variations of this. Oftentimes this will depend upon the condition of that bacteria itself, the host cell, or the condition of the environment. So if the bacterial cell is healthy, essentially, actively metabolizing, oftentimes you'll see this lysogenic cycle ensue. If there is damage to the cell, if the environment is not very conducive to long stage storage, essentially, then you might see a continuous active production of infectious particles. This picture here is showing you, we'll talk about it again in chapter 8, but this is how this DNA is integrated. We call this site-specific homologous recombination. It makes sense if you slow it down. Site-specific, that means that this viral DNA doesn't just randomly incorporate anywhere in the bacterial chromosome. In fact, it's very specific to one particular location on that bacterial chromosome. And this is important because we've been talking about how this integration does not necessarily alter the host metabolism or replication. If you didn't have site-specific incorporation, then you would definitely be disrupting genes at some point along the way, and that could damage that host cell altogether. So this site-specific recombination or site-specific integration does not disrupt that host cell, that host bacterial cell genome. At this synapse, which is a region of DNA, you can see that these guys have homology. That means very similar overlapping DNA sequences from one part of that genome to another, from that virus to the host chromosome. So these guys line up in regions of homology. And then recombination just means to recombine these things. In this case, it's kind of interesting because the recombination event leads to integration, and it does not excise anything out of it. So you don't have any DNA from the host chromosome cut out, right? So you have no removal of the host genes. This prophage stage is maintained indefinitely through something called a viral repressor. This is an I guess it is an enzyme. This is a type of an enzyme that physically sits on top of the viral DNA when it's integrated in the host cell, and it prevents the expression of a particular enzyme called the excisase, which is responsible for cutting out that viral nucleic acid. So it's a regulator of a regulator, essentially. But as long as that repressor maintains bound to that viral part of the nucleic acid, then you have the maintenance of that prophage stage. Now, when that cell is damaged, when there's environmental alterations, like for example here, when there's extensive UV damage to the cell, there's something called a SOS repair, and when this is initiated, it eliminates that repressor altogether. And elimination of the repressor of the phage leads to phage induction or excision of that phage DNA. Excision of the phage DNA, and that leads to a lytic infection. The last thing to mention for these lysogenic stages is this concept of lysogenic conversion. And this is the concept that new traits in the bacterial host can be acquired through that phage DNA that's incorporated into that host chromosome. This is done with those temperate phages in the period of prophage stage, right? These are examples of some pathogens that have their significance due to the phage DNA that's encoded in their genome. So Corynebacterium diphtheriae, which causes diphtheria, the toxin itself is actually encoded by phage DNA in the host chromosome. Clostridium botulinum, the toxin for the botulism, the damaging part of that botulism infection is actually phage DNA. So these are all examples of lysogenic conversion. We are not going to cover transduction in this chapter, so you can sort of put a little check mark on this or bookmark on it. We'll get back to it in chapter 8. Bacteriophage therapy, we'll talk about that a little bit more downstream. This is a really cool concept where you can use bacteriophages to actually treat human bacterial infections. You can think about that in the context of what we've just discussed here and see how beneficial this might be. But we'll come back and talk about this in our chapter on bacterial control methods, so antibiotics or our drugs chapter 10, and then we'll also talk about it during our immunology section. We haven't covered genetic engineering yet, and we haven't covered transmission. So all of these things are in future chapters.